Welcome back to the Deep Dive. Today we're cracking open one of the most fundamental questions in biology, and we're doing it with a really fascinating stack of sources that cover, well, everything from single cells all the way up to us.
It really is the ultimate why bother
Huh.
question, isn't it? I mean, we talk about how organisms maintain themselves, they breathe, they eat, all of that's necessary to keep the individual alive.
Yeah.
But reproduction,
Right. It costs so much energy, and an individual organism doesn't actually die if it never reproduces.
so why do it?
Hmm.
And the core insight from our material is that reproduction is how life makes its presence known. It's how a species stakes its claim. Huge numbers of similar individuals are what really define a species in the world.
And our mission for this deep dive is to get into the mechanism behind that, that claim staking. We're talking about the ultimate blueprint, DNA, and how the inherent flaws in copying that blueprint are actually the engine that drives all of life.
We're going to track that idea from a simple cell splitting in two all the way to, well, the complexities of flowering plants and human anatomy. It's quite a journey.
Okay, so let's start at the very beginning, the blueprint. If body designs are similar, the blueprints have to be similar. And that information, that blueprint, is in our DNA, deoxyribonucleic acid, tucked away in the cell nucleus.
Exactly. And DNA basically just holds the code for making proteins.
Yeah.
And those proteins do, well, everything. They build your muscles, they run your metabolism.
So if you change the information in the DNA, even just a little bit,
You change the proteins. And if you change the proteins, you eventually start altering the entire body design. So the most basic event in all reproduction is simply creating a copy of that DNA. It's a chemical reaction.
and you can't just copy the DNA, right? You have to build a whole new house for it. You make 2 copies of the blueprint, pull them apart, and build an entire new cellular structure around that second copy. 1 cell becomes 2.
And this is where it gets really interesting. And it's something the sources highlight again and again. No biochemical reaction is perfect. It's just not possible.
Not absolutely reliable.
Not at all. So the copying of DNA will always involve tiny variations. The new copies are similar, sure, but they are never perfectly identical to the original.
That's wild. So the very mechanism that's supposed to ensure continuity also guarantees constant change. I guess some of those copying mistakes must just be catastrophic for the new cell.
Oh, absolutely. Some variations are so drastic the cell just dies immediately. The blueprint is too corrupted. But the ones that survive, they're subtly different. And this built-in tendency for variation is, well, it's the basis for all of evolution.
But that sounds like a terrible feature, not a good one. Why would you build a flaw into the most important process?
Well, it's that classic evolutionary trade-off. In the short term, you want perfect copies. If you're perfectly adapted to your little slot in the ecosystem, your niche, then consistency is great.
But niches don't stay the same.
They never stay the same. The temperature changes, a new predator shows up, a meteorite hits. If your entire population is made of perfect, identical copies, the whole species could be wiped out by one event.
But if you have variation,
If you have variation, a few of your individuals might, just by chance, have what it takes to survive. Think of bacteria in temperate water. The water heats up, 99% of them die. But that 1% that happens to be heat resistant, they survive, they reproduce, and they save the entire species.
the individual loses, but the species wins. That makes a lot of sense. So with that in mind, this balance between consistency and variation, let's look at how organisms do this asexually. Just one parent creating the next generation.
And the method they use really depends entirely on their body plan. For single-celled organisms, it's fission, just simple cell splitting. The most basic form is binary fission, like in bacteria, just splits in two.
What's amazing, though, is how a little bit of structure changes everything. A simple amoeba can just split in any direction it wants. But the source material points to something like leishmania.
The parasite that causes Kala-Azar.
Right. It has this little whip-like structure, and suddenly the splitting has to happen along a very specific, definite orientation. It's not random anymore.
You have to make sure both new cells get all the necessary parts. But you can also dial up the efficiency. So instead of one cell becoming two, some successful parasites, like the one for malaria, use multiple fission. One cell just explodes into many daughter cells all at once. It's a numbers game.
Okay, moving up in complexity a bit. Simple multicellular organisms like Spirira, that filamentous algae, they use fragmentation.
It's very direct. It just matures, breaks into pieces, and each piece grows into a new individual. Simple as that.
But that brings us to that complexity hurdle you mentioned. Why can't a human or even a fish do that? You can't just break off a piece and grow a new one.
Because we're not just a simple collection of cells. We have specialized tissues and organs, and everything has a specific place. You can't just regenerate a whole person from, say, a piece of a lung. It just doesn't work that way.
So for complex organisms, asexual reproduction has to get more specialized.
Exactly. You get things like regeneration, in animals like planaria. If you cut them into pieces, each piece can grow into a whole new worm. But that's usually a response to being injured. It's not their normal way of reproducing.
But some organisms have weaponized that ability.
Right. An organism like Hydra uses those same regenerative cells, but on purpose. It grows a little outgrowth, a bud, at a specific spot. When that bud is mature enough, it just pops off and becomes a new Hydra.
And this brings us to plants, because they have taken this to an incredible level, especially for us in agriculture. Vegetative propagation.
Oh, it's a huge advantage. You can grow new plants from roots, stems, leaves. You just bypass the whole seed process. We do it all the time with roses, grapes, even potatoes with their little notches or eyes.
And the benefits are massive, aren't they? The plants grow so much faster, they produce fruit earlier, and you can even grow plants that can't make seeds anymore.
Like bananas or some types of oranges and jasmine. Plus, they're all genetically identical, so you can preserve a really desirable trait, like a particularly sweet grape.
We've even taken that to the extreme with things like tissue culture.
You can take a tiny cluster of cells from the growing tip of a plant, put it in this artificial medium, and it grows into a lump of cells called a callus.
Just a blob of potential.
A blob of potential. And then you add hormones to tell that blob to grow roots and stems and leaves, and you've got a perfect new plant. It's incredible control.
And just to round out the asexual methods, there's spore formation, like on bread mold.
Right, rhizopus. It's basically just packing up reproductive cells into these tiny, durable spores with thick walls. They can survive almost anything until they land on a nice, moist surface and start to grow.
So all of these methods are great for making lots of copies, fast.
Yeah.
But they're all limited by that slow, random DNA copying error rate for variation.
Which is fine if your environment is stable. But if it changes too fast, you might not generate the right variation in time to save the species.
So we need to speed things up, and this is the big evolutionary leap, the invention of sexual reproduction.
This is a total game changer, because now you're not just waiting for random errors, you're actively shuffling the deck. You're taking all the variations that have already built up in two different individuals and combining them.
Creating a totally new combination.
A novel combination every single time. It dramatically increases the odds that someone in the next generation will be equipped to handle whatever the environment throws at them.
Okay, but hold on. If you just combine the DNA from two individuals, wouldn't the offspring have double the amount of DNA? That sounds like it would break the whole system.
That's the huge problem that multicellular life had to solve. And the solution was brilliant. It was specialization. Organisms developed special lines of cells called germ cells in specialized organs.
And what's so special about these germ cells?
They go through a very unique type of cell division called meiosis. And its entire purpose is to cut the amount of DNA and the number of chromosomes in half.
So when the two facets, one from the male germ cell, one from the female, fuse together during fertilization.
You're right back to the original correct amount of DNA. Problem solved. And you get all the benefits of mixing up the genes.
And that specialization goes even further, down to the germ cells themselves, the gametes. They're not identical.
No, not in complex organisms. The female gamete, the egg, is typically large. It's not motile. And crucially, it stores all the food the new embryo needs to get started. The male gamete, the sperm, is tiny and motile. It has one job: deliver its half of the genetic material.
Let's see how that architecture plays out in, well, one of the most visible examples out there. Flowering plants, the angiosperms.
In a flower, you've got the stamen, which is the male part, and the pistil, which is the female part. The petals and sepals are really just, advertising and protection.
And some flowers are unisexual, like a watermelon flower will have either stamens or a pistil, but not both. Others, like hibiscus, are bisexual and have both parts.
On the male side, the stamen makes pollen grains, which carry the male germ cells. And on the female side, you have the pistil. It's got the sticky stigma at the top to catch the pollen.
The style, which is the stalk, and then the ovary at the bottom, which holds the ovules. And inside the ovules are the egg cells.
So you get this two-step process. First is pollination. That's just getting the pollen from a stamen to a stigma. Could be in the same flower, or it could be carried by wind or an insect to a different flower.
And once it lands, then you get fertilization.
Right. The pollen grain grows a little tube. all the way down through the style until it reaches an ovule in the ovary. Then the male germ cell can travel down that tube and fuse with the egg cell. That creates the zygote.
And what happens after fertilization is all about survival and dispersal. The zygote starts dividing, becomes an embryo, and the ovule around it hardens into a tough coat. That whole package is the seed.
It's the future plant, all packed up and protected. And at the same time, the ovary surrounding the seed or seeds starts to ripen and swell, and that becomes the fruit.
And the fruit is really just a clever delivery vehicle. It protects the seed, or it tricks an animal into eating it and carrying the seed somewhere else.
It's a fantastic system, just brilliant evolutionary architecture.
So now let's turn to the species we're probably most familiar with us, humans. And this all starts with that big transition, puberty.
Puberty is this fascinating period where general body growth actually slows down, and that frees up a huge amount of energy and resources to allow the reproductive tissues to finally mature.
And all the changes we see, hair growth, skin changes, breast development, they are, at a biological level, signals. They're external signs that sexual maturation is happening.
If we look at the male system, you have the testes, which produce sperm, and the hormone testosterone. And the sources make a key point here. The testes are located in the scrotum, outside the main body cavity.
And that's not by accident.
Not at all. Sperm formation actually requires a temperature that's a little bit lower than normal body temperature. It's a very sensitive process.
And that sperm, which is mostly just a package of genetic material with a long tail, travels up the vas deferens and gets mixed with fluids from the prostate and seminal vesicles. That provides nutrition and helps it move.
Then you have the female system. The ovaries contain all the eggs a female will ever have right from birth, but they're immature. At puberty, they start to mature, and typically one egg is released each month. It travels down the oviduct, or the fallopian tube.
And heads toward the uterus, the womb.
Exactly. So for fertilization to happen, sperm have to travel from the vagina up through the cervix, through the uterus, and into the oviduct to meet the egg. If that happens, the fertilized egg, the zygote, travels back to the uterus and implants itself into the uterine lining, which has become thick and rich with blood vessels in preparation.
And once it's implanted, this is where we see another piece of incredible biological architecture. The placenta.
The placenta is amazing. It's a disc of tissue that embeds in the uterine wall, and its whole job is to be a lifeline. It lets nutrients and oxygen pass from the mother's blood to the developing fetus, and at the same time, it takes waste products from the fetus back into the mother's blood to be disposed of.
A temporary disposable organ built for one specific critical job over 9 months.
Exactly. And that whole period of gestation ends with rhythmic contractions of the uterus during childbirth.
But what happens most of the time when fertilization doesn't occur?
Well, the egg only survives for about a day. So that thick, spongy uterine lining that was so carefully prepared to nourish an embryo is no longer needed.
So the body just breaks it down.
It dismantles the lining, and it exits the body as blood and mucus. And that process is menstruation, which happens roughly every month.
This all brings us to a really important area: reproductive health and all the societal issues around it. Our sources are clear that just because someone is sexually mature, it doesn't mean they're ready for a sexual relationship or for raising children.
No, these are very difficult choices, and they're often surrounded by all kinds of pressure from friends, family, and even society at large.
And the physical act itself, because it's so intimate, it creates a pathway for diseases, for STDs. You have bacterial ones like gonorrhea and syphilis, and viral ones like HIV/AIDS.
And condoms, as the sources note, can help prevent the transmission of many of these, though, of course, no method is ever 100% effective.
Then there's the issue of avoiding pregnancy. The material lays out a few clear categories of methods. You've got mechanical barriers like condoms, which just physically block the sperm.
Then there are chemical methods like hormonal pills, which alter the body's hormonal balance so that an egg isn't released in the first place, but those can come with side effects.
There are also IUDs, intrauterine devices like the copper T, which are placed in the uterus to prevent implantation. And finally, you have surgical methods blocking the vas deferens in males or the fallopian tubes in females,
which are very effective long-term, but any surgery carries its own risks, like infection.
And the sources bring up a serious societal concern linked to reproductive technology, which is the misuse of prenatal sex determination for illegal sex-selective abortions.
Female feticide. It's a deeply troubling issue, and the fact that it's causing a measurable decline in the child-sex ratio in some parts of the world, even though it's illegal, shows you this dangerous intersection of biology and culture.
And that kind of leads to this final big picture point on population. There's a common argument that our expanding population makes it harder to improve the standard of living for everyone.
But the sources suggest we should pause and think about that. It's possible that inequality within our societies is actually a much more significant reason for poor living standards than just the raw number of people.
So when you pull the camera back and look at this whole deep dive, from a tiny error in a DNA copy to the complexity of a flower or the human placenta, the whole story is really one of balance, isn't it?
It's this constant, delicate tension. You have to copy the blueprint accurately enough to maintain the species in its current niche.
But you also have to copy it inaccurately enough. You need that variation to make sure the species can survive when that niche inevitably changes.
That is the core takeaway. So here's a final thought for you to chew on. Humans spend an enormous amount of energy on our reproductive systems. 9 months of gestation, the hormonal shifts of puberty, the decades spent raising children. Given all that, what societal feature, if we really addressed it, would determine whether our species is actually successful at providing a high standard of living for all the individuals it produces? Is it about controlling population size, or is it something more fundamental about how we share the resources we all generate? Something to explore on your own.
Fascinating stuff. We'll see you next time.